I'm shouting, hoping that Husain's mic will pick me up for the people on Zoom.
We can hear it.
Oh, excellent. Thank you. So it's my pleasure to introduce Husain Poonawala here today to give us a seminar called Developing a Mycobacterial Research Agenda. So it's a topic that I'm mildly interested in. But he's a new clinician here at Tufts Medical Center. He comes from a really rich educational background in terms of both obviously clinical education, or obviously becoming a clinician, but also from a molecular epidemiology viewpoint, mostly focusing, I would say, on TB, doing a lot of work in India and now in New England, and has a real appreciation not only of what's going on with TB disease on the ground in the lab, but also with other mycobacteria as well, and is already starting to integrate himself into the research programs happening on both sides of the street here, but also across Boston, where there's a lot more discussion now of how do we handle mycobacteriology in the clinic and in the lab across Massachusetts. So we're really looking forward to your lecture.
Thank you. All right, so thank you all for taking the time to join in. And so I am appointed to the Division of Infectious Diseases, and I also am one of the assistant lab directors of the clinical micro lab. So the goal of this talk is, I started with med school in India where I really didn't do any research, or we didn't have a lot of opportunity to do research. And now I'm sort of here at Tufts. I have a joint appointment in ID and micro with a research interest in mycobacteria and bioinformatics. And I've done clinical work and research work related to mycobacteria. And I just want to talk about the work that I've done previously that has gotten me to where I am today, and how that's shaping what I want to do going forward. And then hopefully start collaborations with members of the audience. So if stuff you see that I discuss, hopefully some of it's going to spark your interest, and you may have ideas that you'd like to work on with me. So that's the other goal. And so how did I develop an interest in TB? So there's actually no infectious diseases specialty in India the way it is in the US. And so this is the leading causes of death due to infectious diseases. This is on the, on the left-hand side is leading cause in 1919, and now it's like 29 years later. And you can see that basically there's a lot of infectious diseases in India. And if you just look at the number one, diarrheal diseases, 1 million people died of diarrheal diseases in 1990. And 29 years later, there's 600,000 people dying every year of diarrheal disease. So this is just the mortality due to infectious diseases is a lot. And so going to med school in India, you just see infectious diseases all the time, and you don't really think about developing a specialty. It's not a specialty. There's no jobs for infectious disease itself. So when I came to the US, it's when I realized infectious disease is its own specialty. And when I came, before I started residency, I stayed with a cousin, and she sort of got me this book. She had this book on her shelf. It's by Paul Farmer, who sort of done a lot of work on MDR TB and actually changed how we think about treating infectious diseases in the developing world. And when I was interviewing for my infectious diseases fellowship, I didn't really have a clear sense of what infection I wanted to focus on. And so at one of the interviews, somebody says, "What are you going to focus on?" I said, "Well, I like all of infectious diseases." And she's like, "You can't say that, just pick one." So I picked tuberculosis. And so this is a map from 2016. This is the tuberculosis incidence in India in 2016. And tuberculosis is a social disease. It reflects poverty, it reflects living conditions, et cetera. And so if you look in the south of India, that's actually the highest sort of human capital development in India. So they have a very low burden of TB. And so I went to the state that's somewhere in the middle. I don't know if this pointer works. It's called Chhattisgarh. It's sort of the one that's at 45 degrees in the center. And so the reason I went there was I had heard about these doctors working there. They had all graduated from the best medical school in India, and they spent a couple of years traveling around the country. And they said, let's go and find the most backward place in the country to set up a hospital. And so that's what they did. So when I was a resident, I went there. And so they did everything. They did complex pediatric surgery. They took care of patients with tuberculosis. They had a leprosy clinic. And so I went there for a few weeks as a resident, and I got to sort of take care of patients just to see what it was. And one of the things that's really interesting or sad over there is the degree of malnutrition, where we would see people walking in with body mass indices of like 18, 19, 20. And so the first day that I got there, I took care of this patient. And I think the reason I wanted the slide is so TB is both under and overdiagnosed in India, and that reflects sort of why I decided to focus on the microbiology part of it. So this is a 22-year-old who came in. She'd been seen maybe a couple of years ago, and at that time she weighed 37 kilos. But now she came in with a month's history of cough, shortness of breath, classic TB, but she weighed only 26 kilos. So her BMI was probably around 13. And she came in and she was hypotensive. So her blood pressure was like 70 over 50 is what I have looking back at my notes. So we treated her with fluids but she didn't do very well. And she passed away very quickly the next day. And she had sputum that was positive for AFP. So she had TB. This is her chest x-ray. But then the question that came up is like, well, TB divides only once every 24 hours. What made her sick so quickly that she passed away? And how do people actually die of TB? When you say people are dying of TB, there's 1.5 million people who die every year. What is the biological endpoint? Do they have adrenal insufficiency? Do they have, superimposed bacterial infections? What is happening? And this is a place where we would want to answer this kind of a question because they're seeing these kinds of patients, but they don't have the lab capacity to do that. They don't have automated blood cultures. They don't have, trained technologists to gram stains. We don't have great automated susceptibility testing. So we don't exactly know what's happening. The other patient that I saw was actually somebody who came in with a blast crisis. So a blast crisis is when you have leukemia that's full blown, and his white cell count was about 30 or 40,000. And so I was wondering, why has he come in so advanced? And let me look in his chart a little bit. And he had come a year ago with anemia, with symptoms of anemia, and his white count was his hemoglobin was about 10. In the US, he would have gotten a bone marrow and they would have diagnosed acute myeloid leukemia and they would have started him on treatment. But in this situation, everybody who comes in with this picture, often they think of TB. So he got treated for TB, even though he didn't have TB, when he actually had a leukemia that needed to be diagnosed and treated. And so when we talk about tuberculosis as a public health issue, it's not just about diagnosing and treating tuberculosis, but it's also the fact that you need the entire diagnostic setup to diagnose what the patient has, and that has its own interventions. And so that's sort of why I ended up deciding that I wanted to focus on micro. And so as a quick sort of refresher, so AFP, you can do smears, which are not very sensitive. You can do culture, which takes a long time, usually about 6 to 8 weeks in a clinical lab. And you can do automated bacterial cultures, liquid cultures, which is much more sensitive. It's faster, but it's expensive. And so the estimate is that only 1% of TB in India actually gets cultured. Almost all of the TB in India is diagnosed by smear. And that's common in most of the world as well. So this is the Gene Expert, and I think this is an automated test. The best description I've heard is that think of making coffee in a K-cup. You take your K-cup, you put it in the machine, and you're going to get your result. You're going to get your coffee really quickly. And so this is by actually David Allan's lab in New Jersey developed the test. And so it's a very simple system. You take the sputum, you liquefy it, you put it in this little cartridge, and it'll give you two answers. Is there TB or not? And is there rifampicin resistance or not? And I put game changer in quotes because that was like all the editorials about this when it came out, they used the word game changer. 10 years later, it looks a little different. It's not had the impact that we would want it to or we thought it would have, partly because healthcare systems are not perfect. So you can diagnose the TB, but if you don't have a system to treat and follow the patients, you're not really going to reduce mortality. And so my project, so I'm going to talk a little bit about the projects I did when I was a fellow. So we used the Gene Expert to actually see if we could take people out of airborne isolation if we thought they had TB. So when patients come into the hospital, if we think they have TB, we put them in isolation. So that's a negative pressure room so that they don't infect anybody or they don't expose anybody. And so the standard used to be you get 3 AFP sputums, and then if they're negative, you take them off precaution. So that would mean that patients would often stay in isolation for 3 or 4 days, sometimes longer. So we wanted to use this test to see, could we do this faster? So could we just do this in a day or two? And so we had about 300, so this was a prospective study. We had about 300 patients who were isolated. We were able to we had enough sample from about 150 of them. And ultimately we analyzed 131 of those episodes. And so what was interesting was in our demographics was that we had a lot of risk factors for TB. This was in Baltimore. So 2/3 of our patients were immunosuppressed, either HIV or chemotherapy or transplant. And there was a fairly significant history of homelessness, incarceration, and TB. But what was interesting is we actually didn't find a lot of TB in our patients. We only had 3 patients who were diagnosed with TB, but we had a lot of NTMs. So about 10%, 8% of our patients were ultimately diagnosed with non-tuberculous mycobacteria, so either MAC or Kansassia gordonae or abscessus. And I thought that was very interesting to me was that, you know, we keep thinking about TB, that's what we're sort of drilled into us as medical students. But when I'm doing this study, I'm not finding that much TB, even though I have all these risk factors for TB. And what I think is interesting over here is that the positive predictive value of AFP smear for TB was only 20%. So if you had an AFP smear that was positive, there was only a 20% likelihood you'd have TB. And that's because most of our patients had non-tuberculous mycobacteria and not TB. And so this sort of clinically becomes relevant because then, I'm going to skip this, but what we did show is that when we implemented this test, if we were to we ran a couple of scenarios, if we were to just implement this test, what impact would it have on isolation? So the standard was, it was taking people about 3 days of isolation. And if we just use the test without sort of any communication with the provider, so that's the dark line which goes along. So this is a Kaplan-Meier curve. If we use just use the test instead of smear, and we reported the result, we'd reduce the isolation to about 2 days, so 42 hours. But if we introduce the test and then we call the clinician and we told you, hey, the smear is the expert's negative, you can take them out of isolation, we'd reduce it even further. And the point I'm trying to make with this is basically that it's not just about introducing a rapid test. You have to have the systems in place where if the lab runs the test, somebody needs to be able to act on it. And so that's what we showed with this test. And if we looked at how much isolation would we save with this, we observed for our patient population, we had 378 days of isolation. Depending on how we reported the result, we could get that down to 82 days of isolation for all of these patients. And we also noted there was a big delay. So even with patients who had a negative smear, they were being kept in isolation on average for 24 hours extra, even when they didn't need to be. Patients who had NTM infections were isolated for 147 hours, so almost 6 days. And so what I think this sort of told me is that there's actually a declining burden of TB in the US. And this is the most recent data from the CDC, where in 2020, we only had 7,000 cases of TB. But what we do have actually is a lot more NTM. And so how I got interested in NTM is the lead author on this, Paul Salib, was actually one of the faculty members where I was a fellow. And he at the NIH validated the MALDI-TOF, which is what we use in our clinical lab routinely. He validated the use of MALDI-TOF for diagnosing non-tuberculous mycobacteria. And so he told me, you know, if you're interested in TB, that's great, but why don't you think about NTMs as well? And he told me to do a rotation at National Jewish in Denver. So National Jewish is in Denver because it's the Mile High City. And traditionally, when we didn't have treatment for TB, you put people in high altitude sanatoriums. And it started out as a TB hospital. And then now over time, they've evolved into an NTM center. And not only do they take care of patients with NTM, so I had a 4-week rotation over there where we had new patients come with NTMs and we'd evaluate them, but they also have a reference lab to diagnose NTM infections. So a pretty interesting experience. And I think, again, it sort of said, you know, there is more to mycobacteria and got me interested in it. And so the burden of NTMs is increasing in the US. So this is a recent slide. And I think the biggest risk factors, if you look at both of these is that patients over 65. So there's an increasing proportion of patients over 65 who have NTM infections over time. So just age is a big risk factor. And this study actually showed that when they looked across the US, the Southeast had a lot of NTM infections. That's Florida, Georgia, Arkansas, Mississippi. And the annual burden of NTM infections is in 2010 was close to $1 billion a year. That was how much it was costing people. And the highest burden of NTM in the US is actually in Hawaii. And so this is from the island of Oahu, where they did, they sampled geographically, they looked at where patients were from different parts of the island, and they calculated the burden of NTM infection. And the burden of NTM infection in Hawaii rivals that of TB in many parts of like middle and high burden countries. There's just so much NTM there. And so the question is, what is it about NTMs that do this? So when we talk about non-tuberculous mycobacteria, there's actually about 186 species. So there's a lot of NTMs out there. and they like watery environments, lakes, rivers, soil. It's not really clear how humans get infected. There are a lot of theories out there. People say aerosolization, shower heads. Gwen Hewitt, who's at National Jewish, she was obsessed with sort of saying acid reflux was how most people were getting NTM infections, because she said NTMs are in the water, and if you have acid reflux, that spills into your lung, and that's how you're getting NTM infections. We actually don't have great data on NTMs, partly because it's not a reportable disease. So the US has really no registry to track NTM infections, but they're also very hard to grow and identify. So if you don't pick up the NTM or you don't diagnose the NTM, you're not going to then realize what the burden is. So that's sort of the catch-22 situation over here. And the diagram on the right is just sort of, NTM infections are a complicated pathophysiology. There's host factors, there's environmental factors, and there's intrinsic organism factors. So you can have a variety of immunosuppression that is either acquired or you're born with. You also have these pathogens that have these really resistant cell walls, which are thick. Mycobacterium abscessus is resistant to bleach. I mean, the cell wall is that thick. They're in the environment, so it's easy to get infected with them. And this was one sort of model of NTM infections is what they say, you have more than one hit. So you could have a genetic mutation that affects your ciliary clearance, or you could have another immune deficiency. You could have a CF mutation, not a complete CF mutation, but partial CF mutation, which have been described as a predisposing factor for NTM infections. Or you could have lung disease. You could have alpha-1 antitrypsin disease, which sort of is a risk factor for lung disease. And then either you get older, or you have structural lung disease, or you have surgery, and something changes and now you're at risk for NTM. So the point that they were making is that it's not one risk factor, but it's a confluence of risk factors that increases your risk for NTM disease. This is a paper from 2017, and it sort of goes, they used long read sequencing on common NTM species. And it's sort of, I think of NTM of mycobacteria as a spectrum. So right at the bottom here, this is working. So at the bottom in the red, you have Mycobacterium abscessus, which is sort of the hardiest of the mycobacteria. It's the most intrinsically resistant. And as you go clockwise, you go from the slow-growing mycobacteria from the fast-growing mycobacteria to the slow-growing. And you end with tuberculosis, which is in gray. And so tuberculosis is very different because there is no environmental reservoir of tuberculosis. It's only humans. You cannot get it from the environment. And so over time, what they sort of demonstrate is that mycobacteria have lost DNA, and they've evolved from an environmental pathogen to an intrinsically obligate pathogen. So for example, the genome size of Mycobacterium abscessus is about 6 megabases. The genome size of TB is only 4 megabases, and it's lost a lot of that DNA along the way. And so there's a lot of genetic variability among the mycobacteria. And they also cause a very, very wide range of infections. So you have all of these species, you have a bunch of different infections, and the treatment is not the same for all of them. Depending on the species, your infection is going to change. So if you have Mycobacterium avium complex, for example, you could get disseminated disease if you have HIV, or you could be a little old lady who has isolated lung disease. If you have sort of Mycobacterium marinum, is commonly seen in people who handle fish tanks. So your predisposition sort of to each of these infections changes. And they're really, really resistant. So these are the MICs for tuberculosis. So isoniazid, it's 0.1, rifampin is 1, ethambutol is 5. If you look at Mycobacterium abscessus, the MIC, or the concentration of antibiotic required to kill abscessus, is over 512. So that's a 5,000-fold difference. And so that is sort of where, it's adapted, the NTM start off as environmental pathogens that have all of these cell wall mechanisms as well as other mechanisms to be resistant to other bacteria which are probably producing the antibiotics. Then you end with TB, which is now evolved to only be a human pathogen, and it's lost a lot of the DNA that also confirms its resistance mechanisms. And so that's why it's a lot more susceptible. And there's both acquired resistance and there's intrinsic resistance on a variety of different levels, different mechanisms. It could be mycolic acid, it could be peptidoglycan, it could be ribosomal, it could be a fluoroquinolone action. So there's a lot of different ways that each NTM species can be resistant to the antibiotic you want to use. And I think of Mycobacterium abscessus, which is, it's getting a lot of attention. It's sort of very popular. It's not the most common one, but it is the nastiest of the NTM infections. And I would probably say it's worse than MDR TB in some ways. And these are sort of the treatment recommendations is sort of, your initial treatment, you need 2 to 3 IV drugs. You need 2 to 3 oral drugs. So you're telling somebody you're going to need 4 or 5 drugs. Each of these is not, amikacin on its own is toxic. You're saying you want to add amikacin, then you want to use linezolid, and you want to use clofazimine. So these are nasty drugs, which are not pleasant individually. And now you're telling people you need to take 4 or 5 of them together for 4 to 5 months initially, and then potentially for at least a year. And the average, so the next slide goes over sort of, this is a patient level meta-analysis where they looked at all the publicly available data of abscesses, and they looked at treatment outcomes. And so the ones I boxed in red are the ones to focus on. So if you look at the total number of patients, the success rate was about 40%. And if you divided the abscesses by subspecies, there's abscessus abscessus, which is one of the subspecies where your success rate was like 30%. And if you look at how long people were treated for, we're talking about 2 years of treatment, at least for most of them. So I've told you about all these toxic drugs that now you need to treat people for about 2 years, including IV drugs. And some of them needed surgical resection because they weren't getting better with medical treatment. So they said, let's go and take your lung out. Maybe that's going to fix things. And so I had all of this exposure to this interest in NTMs. And then so I said, let's do a microbiology fellowship because I think based on my experience in India, I think microbacterial diagnostics are important. And then I realized that, the NTMs are a wild west. We do need to sort of diagnose them better. And so this is the Winchester revolving rifle. And so I went to Yale for my fellowship. And so Winchester, the gun was actually invented. The guy who founded the company was in New Haven. And his son died of TB at the age of 22. So his wife sort of gets obsessed with TB, and she builds a TB hospital in New Haven, which gets bought by the government and is now a VA. If you look really carefully on one of those towers, you can actually see the VA over there. So that's the VA hospital, which originally used to be a TB hospital. And so my fellowship was actually named the Winchester Microbiology Fellowship because the Winchester family paid for it. And the VA also has a microbacteriology reference lab. So I got to sort of learn a lot of the clinical techniques used in the diagnosis. And so as a fun fact, Sarah Winchester ultimately goes to San Jose, and the story is that she's so haunted by the deaths that the rifle was responsible for and that the money she made for it, that she built this house and she kept adding rooms to hide from all the ghosts that were coming after her. And if you want to watch a movie, that's Helen Mirren, actually, who stars playing the role of Sarah Winchester. And so we have platrons in the micro lab. And sometimes people wonder, are we really accomplishing anything? So this was an outbreak that was reported in 2017. And I had a role in it at Yale because one of the techs actually told us, she comes to me and says, hey, we have this wound culture. The gram stain looks like there are branching gram-positive rods. So I said, okay, this might be a mycobacteria. Why don't we subculture it on the mycobacterial plates? So we did. And then a couple of days later, the resident calls me and says, hey, that plate you were talking about, that's positive for this growing Mycobacterium abscessus. And then she looked in the chart and said, this patient's had plastic surgery recently. And so I had heard about this previously, that people who go to the Dominican Republic, it's a very known risk factor to get skin and soft tissue infections after plastic surgery. So I said, let's talk to the fellows about this. So I talked to the fellows about this. So we have platrons in the micro lab. So that's when we take the actually interesting cases and we teach the fellows that, hey, this is what the plates look like. This is the testing we do. So I talked about it in rounds. And then later that evening, the fellow got a consult for another patient with a skin infection after plastic surgery that wasn't getting better. And both these patients were best friends who had gone together for this infection, for surgery. And so this obviously went to the state public health department and then reached the CDC. And then they did a multi-center investigation. And they found this one place that these patients had surgery for. So what they did was they did whole genome sequencing of all of these isolates. And what was interesting was that they found there were 22 cases. They had 3 distinct genetic cluster variants, but none of those clusters corresponded to a single clinic or a single surgeon. So we know that these people went to this place to get plastic surgery. We know that's a risk factor, but we can't prove it, using whole genome sequencing. And that raises questions about the diversity of NTMs. And if we do whole genome sequencing, what do we need to sequence, like just the core genome, the accessory genome, and how do we make sense of all these findings? Are we using the right sequencing technique? Should we be using long read sequencing? So a lot of questions about when you do whole genome sequencing and try to do outbreak investigations, you may not always get the answers, even though something's telling you they all got the infection in the same place, but you can't prove it. And so there was another, so I mentioned the Yale Mycobacterial Reference Lab. So we had this case that we ultimately published as a letter to the editor. The TB grew, the mycobacteria grew in our liquid culture, and it cross-reacted with an AccuProbe. So that's a commercial probe. And it cross-reacted with Mycobacterium avium complex. But when we looked at the plate, it was yellow, which is a scotochromogen. So it shouldn't, the cross-reaction is also false positive. We used MALDI-TOF, which is this new fancy thing, and it gives us a different species, Mycobacterium gordonae. And then we had to sequence it with the 500 base pair ribosomal RNA sequencing. And we got these three species, Mycobacterium parentium, Malmoense, and Salatum. Two of those were not possible because this was a scotochromogen. And I think what I'm trying to sort of highlight is that identifying the mycobacteria, even in a reference lab, is not straightforward. You need to have a bunch of different techniques, and you need to be able to sequence, and you need to be able to sort of look at the plates, look at your sequencing results, and come to it. And this was, I think, the third report of this bacteria ever. There was one from Brazil, there was one from, I think, South Africa, and this was the first one from the US. And so there's probably a lot more mycobacteria out there that we're not picking up, either because the specimens are not in a reference lab or the labs don't have the ability to do this level of identification. And so after I finished my fellowship, I went back to India to do research. The research project I actually wanted to do, I couldn't do because the machine that I was told was working was not working. And a year later, when I left, the machine was still not working. So while I was there, I said, why don't I do something else? And I worked with, so Narendra was a postdoc at Cambridge. Sharon Peacock was his mentor, and she actually is still at Cambridge, and she ran the UK's sequencing for COVID. She was the director of it. And so he was his postdoc. And so the two of us were there. And we said, why don't we look at spoligotype data and let's try to understand how diverse TB is in India. So when we say Mycobacterium tuberculosis, we're actually saying Mycobacterium tuberculosis complex, because it's not a single pathogen. It's a bunch of them that are very closely related. And so starting at the top is Mycobacterium cannedii, which is also called the smooth bacillus, because when you look at it, it's smooth. And tuberculosis is tubercle because it's rough. And so there's been an evolutionary shift from cannedii to tuberculosis, and that they say that that's also responsible for the virulence. There's only a handful of cases reported of cannedii, and most of them are in the Horn of Africa. But then you have what they call the modern TB and the ancestral TB lineages. You have Mycobacterium africanum. And what the RD and the TBD basically is what I said earlier, is that TB keeps losing DNA. And as it loses DNA, it evolves. And so RD is region of deletion. So each of these regions of deletions is a marker for which subspecies it is. And so this is a fantastic paper where they basically show that humans and TB both emerged out of Africa. So I mentioned sort of the animal strains, and then you have lineage 5, 6, and 7, which are out of Africa, and then lineage 1, which is mainly in South Asia. So those are what they call the ancestral lineages. And then you have what we call the modern lineages, which are 2, 3, and 4. This paper, basically, they used evolutionary molecular clock methods, and they basically would try to plot how did TB leave Africa at how many years ago from the Neolithic area. So it started with lineage 7 so about 73,000 years ago is their estimate. And as you can see, it keeps branching off. And about 40,000 years ago is when you get lineage 2 and lineage 4. And one of the things that they talk about here is sort of the hunter-gatherer hypothesis, as well as the agricultural basis. So the ancestral strains of TB, the ancestral lineages of TB were basically, they're not that virulent. And the idea is that human density is not, because they're all hunter-gatherers, there's not a lot of human density. So you don't want to kill off everybody. You want to let the TB grow slowly so that there's enough opportunity to infect somebody else. And then with agriculture, what happens is, you have increased density. And at that point, you're not worried about infecting you're not worried about not finding somebody to infect. So now you can infect people quickly, and it's okay if you kill a few of them on the way. And so that's why lineages 2, 3, and 4 are called the modern lineages. And the thought is that they're actually more virulent. And there's a good amount of data actually supporting that, which obviously not going to discuss here, some data actually suggests that lineage 4 is continuing to evolve and become more virulent. And this is also really fascinating, where they basically use a phylogenetic tree of all of the TB strains from each country with the mitochondrial DNA from the most common ethnic groups within a country. And they say that basically they map very similarly and suggest that TB and humans have co-evolved. And this is data from San Francisco, which shows that your ethnicity determines what strain of TB you're going to get infected with. So if you're from East Asia, your probability of getting infected with lineage 2 is far more likely than getting infected with lineages 3 or 4. Similarly, if you're from South Asia, you're more likely going to get infected. So the Philippines, for example, is you're going to get infected with lineage 1. And if you look at the table on the right, if your tuberculosis lineage is East Asian and your Chinese ethnicity, your adjusted odds ratio of getting infected with that lineage is 20 times higher than any of the other lineages. And then this is data from Montreal showing pretty much the same thing. They had more countries, they had 10 more countries, and they showed that, Canada and Haiti are sort of, you get lineage 4, which is the Euro-American. India is East African Indian and Indo-Oceanic. So basically by country, your risk of getting infected with the specific lineage changes. And so you'll see all of these different ways of typing, but they're all very similar. And they all basically say there's 6 lineages of TB, 1, 2, 3, and 4, which are sort of worldwide, 5 and 6 are in Africa. Lineage 1 is what I was mentioning as the ancestral lineage, with the 5 and 6, and 2, 3, and 4 are modern. So what's polygotyping? It's basically using CRISPR. So there's a direct repeat. TB has 43 direct repeats, and you have spaces in between. So you amplify the direct repeats, and then if you hybridize it to a membrane, you can identify the loss of some of these direct repeats. And basically, you can code it. So you have a binary representation, which is 43 ones and zeros, and you can convert that to an octal code, which is only 15 digits. And so there's this, and this is a way to actually type TB. And even though we do whole genome sequencing most places, you can actually use the whole genome sequence data to identify the polygotypes, just using the genomic coordinates. So it's a fairly robust and consistent way of typing TB for epidemiological purposes. And so you can create minimum spanning trees, which is sort of a marker of how related, using these digits, these codes rather, you can actually link, you can cluster related strains together. So if it's lineage 1, it's going to cluster together. If it's lineage 2, it's going to cluster separately. So this is data from Kuwait. So the green is EAI, that's lineage 1. CAS is lineage 3, so that's at the bottom. And then the sort of hodgepodge in the middle is mainly lineage 4. And so this map is wrong. And the reason I say that is this has been published, I think, since the first map was 2006, this paper is from 2018, so over 12 years. And this map is by the same author, Sebastian Gagnon, who's great. But the interesting thing was this map was created using only, I think they used 800 isolates, but there were only 17 from South Asia. And so they didn't actually have enough data from South Asia to create this map accurately. So it's showing lineage 4 in Sri Lanka or South India, which is actually not accurate. There is no lineage. You're not going to see lineage 4 there. And so there's limited data from India, and this is sort of older data showing that there is a north-south divide in lineages. So lineage 1 is more common in the south, and lineage 3 is more common in the north. So we decided, why don't we do this with all the data that's out there? So Naren and I did this over the course of a few months. So we collected all the publicly available data. We put it in a database. We put it in an Excel file. We used the binary and the octal data. And then this is the SITVID2 database. So if you need information on any polygotype anywhere in the world, you can go there, download it. It's freely available. And then we basically threw all of our data in and we tried to identify the polygotypes, which we did. And so what we found was there's a tremendous amount of regional variation, but most of our studies are clustered to the big cities, we had 8,300 isolates and 1,500 patterns, which is only 0.0002% of all the cases of TB during this. So our data is not representative. And we found that about 700 patterns had never been described before. And 11 of these patterns had as many as like 7 to 29 isolates. And we emailed these folks who run the database, and we asked them like, hey, do you have more information about these? They had a couple of isolates, that's it. So they didn't have a lot of this data in their own database. And if we included this data, we would go, so the database at that time had about 9,000 isolates. If we included our data and it was all approved, we'd increase the size of the database by 10%. And we found that sort of lineage 1 and lineage 3 were most common, which is what everybody knew. But we also looked at lineage prediction algorithms that actually did not do very well on data from India. And so they failed, which I think becomes relevant because we're doing a lot of whole genome sequencing with TB, and there's a lot of prediction algorithms being designed on data, sort of try to predict resistance, try to predict virulence, et cetera. And I think if you don't account for the fact that you don't have data from everywhere in the world, those algorithms are not going to work well. And I think the purpose of this is basically just to show that on the extreme left is lineage 4, and right at the bottom is lineage 2. And compared to the other isolates, they cluster very, very closely. And there's just so much genetic diversity in India that we are not capturing. And if you see these pink dots, these are actually all of the spoligotype patterns that we did not know which lineage they belong to. So we create a minimum spanning tree just using the data and say, let's see where this settles down. And a lot of it, as you can see, settles around lineage 1 and lineage 3, which sort of, tells you that we probably are missing a lot of the genetic diversity. And it's not in one pocket or it's not in one area, it's all over. We also showed a lot of regional differences. So if you look at sort of South India at the bottom, it's all lineage 1. If you go to Northeast India, which is really close to China, and ethnically they look East Asian, you see mainly lineage 2, which is what we see in people of East Asian ancestry. And if you go up north, you see a mix of mainly lineage 3, which is what we expect. But we found within this, sort of smaller variations. So there's this one family here. If you look at lineage 1, this family called Manu, M-A-N-U, most of that was actually only in Western India. We didn't see it in the rest of the country. We just saw this in Western India. And we create these minimum spanning trees with sort of isolated lineage 1 with isolated data. And if you look at, on the left is lineage 1, and within each family, there's fairly defined clustering. And in some states, so there's data showing in South India that in one state, you're going to have an equal amount of lineage of EAI3 and EAI5. But if you go to another state, that varies, the proportions vary. And this, I think, reflects that the human migration into South Asia is actually quite complex. And there's a lot of sort of genetic diversity in India. And I think that that may be a way to also understand why you have so much diversity of TB within the country. And if you look at on the right, this is lineage 4. It's the predominant one is what we call the T family. Now, this is part of lineage 4. Lineage 4 is the most common worldwide. Within that, LAM and H are actually the most common families. T is not. Some people don't agree that it's even a real family, but we are finding this in 2/3 of our isolates from India. And when we looked a little bit, this is all linked to West Asia. So one sort of thought about migration into South Asia is that they came from the Central Asia through West Asia into South Asia. And so this probably supports that. And then we try to predict the lineages using these algorithms. And most of our data that we think is lineage 1 and 3, which is consistent with what I've said. And so again, why does all of this matter? Why do we need to know this molecular diversity? Because this is data from the Broad Institute where they looked at, I think, 200 isolates from 1990s. And they looked at the whole genome sequenced them. And what they found was that a lot of the mutations that they were seeing just for isoniazid and rifampin had not been described. And the test I told you about, the gene expert, that is on the premise that 95% of rifampicin resistance is in this one area. Here they found 26% of their rifampicin resistance were outside this area. So you're bringing in this commercial test, which potentially is not going to detect what it's supposed to detect because the diversity of strains in your country are completely different from what this test was made on. And it's not just diagnostics, but vaccinology. If the hypothesis is that TB has co-evolved with humans, your epitopes, the antigens that you're going to use need to take into account that maybe a vaccine is going to perform differently in different parts of the world. And so this is sort of, I mentioned why the Gene Expert, I put game changers. This was a cluster RCT that was rolled out in Africa in 2015. And they looked at the mortality. They compared Gene Expert with microscopy, and they looked at mortality, they looked at diagnosis of TB, and they looked at loss to follow-up. And essentially what they showed that is using this really fancy, expensive test did not improve mortality. And the reason for this was the healthcare system itself was weak. And it's really interesting. Cepheid actually, after this paper was published, Cepheid came out with a disclaimer on their website that our test will not fix problems in your healthcare system. That's really what they said. Because there has been a lot of criticism about the amount of money that has been spent. So in India, one estimate was it would be 25% of the national TB budget if they tried to roll this out to everybody. And Cepheid has made a lot of money because this was their first test. Now they've expanded into everything. They're doing C. diff, they're doing MRSA, they're doing everything. But they actually haven't fixed TB, which is what they were supposed to do. And so this is another study that does sort of whole genome, that looked at whole genome sequencing. In 2017, they looked at 5,000 isolates. And if you look at the graph on the bottom, Southeast Asia region, that's for some reason, Southeast Asia and South Asia count as the same according to the WHO. But even though the burden of TB was highest in Southeast Asia, they had almost no strains from Southeast Asia. So like I said, they're missing a lot of TB in, we're not capturing the diversity that exists. And the same thing exists for NTMs. So if you look at this map it's from 2003, this data from 2008, all of the white is essentially the developing world. You have South Asia, you have most of Africa, where you're not actually getting data about the NTM prevalence. And you see each panel is a different mycobacterial species. So you have Mycobacterium avium, Kansassia, Xenopi. And the prevalence of each NTM changes in each area. So if we think there's a lot of NTM worldwide, if we think it's an environmental pathogen, if we think that it's complex immune susceptibilities, but we're not picking up most of the NTM in different parts of the world. We're losing out. And this is sort of an argument about why you need to not just build diagnostic tests, but you actually need to build diagnostic laboratories that can work where they're needed. And so I'm just going to end with a case that we had on the wards recently. We had a 20-year-old male from a TB endemic kind of country who came with cough, shortness of breath, hemoptysis, and weight loss, which is classic TB. He left his home country just 4 weeks before he came to the US. So obviously TB is top on the differential. He got a couple of courses of antibiotics. This is his chest X-ray, which shows sort of a non-specific infiltrates in both lungs. He had a CT which showed a bunch of findings, basically saying he had an infection in his lung. So he was put on airborne isolation. He got 3 AFP smears. He got the Gene Expert that I talk about. We probed a little bit and we found he has this history of pancreatitis. And he had a pleural effusion, so that's a collection of fluid around his lung. There was a fistula, which is a leak between the pancreas and that cavity around the lung. So he needed surgery for that, and he had a blood clot in one of his veins. He got a CT over here, which showed a massive spleen and showed features of chronic pancreatitis. We did more testing, and the AFP smear was negative. But he grew Mycobacterium abscessus. And this is somebody who is from a TB endemic country. And so, again, I think the other thing to highlight here is that we have Mycobacterium abscessus, which the test is being done at the state lab. The state lab doesn't have the ability to do susceptibility testing, so they send it to Tyler, Texas. And when they get the susceptibility back, you see all of these antibiotics are susceptible only to amikacin. But as I mentioned previously, you need to use 5 drugs. Some of these drugs are really expensive. You can see the $3 next to imipenem and linezolid. Those are sort of the effective drugs, but they're really expensive. And he was quoted, I think, $15,000 a week for amikacin and about $250 for 2 other antibiotics. And he's a student, he's in his 20s. He's just come from his home country, which is in the developing world. So is he going to pay? I don't think I can afford $15,000 a week out of pocket. And most of us probably couldn't. And so the questions that this came up was sort of, where did he acquire Mycobacterium abscessus? We don't know anything about Mycobacterium abscessus in most of the developing world. We think it's an environmental pathogen, but how common is it? Why did he get abscessus? Does he have CF? because he has this chronic pancreatitis. He has these lung abnormalities. It's, I think, a month, and we sent the CF gene testing. The results are still not back. So this is in the US. We still don't know sort of whether he has CF or not, but is that why he got it? He's 24. He doesn't have any risk factors. He's not been exposed. What antibiotics do we give him? We need to give him 5 antibiotics that's resistant. Is it even going to work? Because we know the outcomes are poor. And who's going to pay for it? And so we have, I think his social situations, he's a student living in an apartment. He has no social support. He's here on his own. He's going to need 12 months of treatment with these really toxic drugs, which could either leave him deaf, unable to feel, or weird skin, a lot of problems. And so how is he going to manage? So I think the plans about maybe facilitating that he go back home and study remotely and get treatment at home, where a lot of these drugs are going to be easier for him to get. And then I think, would he have been diagnosed appropriately in his home country? In most developing world situations, somebody who comes in with this picture is going to get treated for TB. He's not going to respond to TB treatment because the pathogen is highly resistant. He's going to be labeled as a failure or a defaulter. He's going to be put on MDR treatment, and he's going to be labeled as a difficult patient. And he may not do very well ultimately. And so my current work is I'm working with Maha Farhat at HMS, where we're looking at transcriptomic data to identify, create a diagnostic test for drug-susceptible TB, and hopefully I can present that at a later time. We're working on a couple of grant applications where we want to look at NTM outcomes, and Bree and Deb and folks, so it's a collaborative effort. And we want to adapt the diamond assay for NTM susceptibility testing. Like I said, it's a wild west. We have no idea what we're doing with susceptibility testing. And then ultimately build mycobacterial diagnostics at Tufts Medical Center, because we're doing a lot of send-outs, but we have a need for it. And I'm going to end with this this quote that I read years ago that I always think about. So it's by Paul Farmer. He says, "I have been working in Haiti for almost 20 years, ever since I was a young chap. And some years ago, I was asked by the state of Massachusetts to be a TB commissioner. I said, what the hell do we do? I was in Haiti and I had a lot of TB patients, and I took sputums and I brought them to Boston. And I took them into the lab and I wrote Paul Farmer, state TB commissioner. I wanted to process my samples from Haiti, and they did and never asked any questions. So I did it more and more, and then I did it with sputums from Peru. And of course, eventually they asked me why. So I said, Massachusetts is a great state. It has a big TB lab, lots of TB doctors, lots of TB nurses, lots of TB specialists. It lacks only one thing, tuberculosis." And so I think big thank you to Deb for hiring me and being very supportive of everything that I'm trying to do. Celeste and the folks at CIMAR, and then Maha, who's I've been working full-time clinically for the last 3 years, and I've been working with her nights, weekend, my time off. She's been very patient as I've tried to get this project to the finish line, and we have. And then ultimately my wife and my 2 kids, who I think have really supported me through a lot of what I've been trying to do. And I think that's it. So questions? Bree. It's a great question. So I asked when I was in India in 2017, I asked this question. And so I spent a month at this hospital that sees a lot of MDR TBs, and they do a lot of culture. When I say a lot, I mean they have 1 or 2 XDR TBs a week and 3 or 4 MDR TBs. So she said that about 1% is what they saw NTMs as co-infection. She didn't see a lot, but I think the tricky part here is your threshold for detection. And what they were doing there is only liquid culture. So if you want to detect co-infection, what you really need to do is be able to do solid and liquid. And on the solid plate, you can pick out the colonies that don't look like TB, and you can actually identify. And so what actually happens in India, and I've seen this, is it's very binary. This is TB or it's not TB. And so if it looks like something that's not TB, they label it as a contaminant, and they don't work it up any further. So I think this is not uncommon in other parts of the world as well. But this is probably why we don't know where the NTMs are. Yeah, it's always been it's an afterthought, right?
In the TB community, why they see that results in TB endemic. same level of protection after. That's the relationship.
I don't know, and I doubt it. I don't think so. I mean, because the pathophysiology is very different. And I think that, so if you look at sort of the test that we use to diagnose latent TB, what do you call it, the quantiferon. So it's a T spot. It's basically a stimulated T cell stimulated with TB antigen that looks to see sort of, are you exposed to TB or not? It doesn't really cross-react with most NTMs. It's only known to cross-react with 2 or 3 NTMs. And so if there truly was a benefit, I would imagine that it would light up all the time if you really had that much exposure to an NTM. So my guess is not. Roberto.
Concentration, because what you try to get information.
Absolutely, and I think the data that we've been doing is looking at publicly available data, and there's a lot of it is mostly H37RV, the type strain. Maha has used this data to submit for an R01, where we hopefully will actually answer that specific question. I think we'll just use what we do in a clinical lab. You know, what are the concentrations we use for TB testing in a clinical lab? What are the antibiotics we use? And start with that. And I think if that works, then we'd expand to sort of different strains and different antibiotics. But I mean, the data, hopefully when I can present it, is fairly compelling that this is viable. And we're getting results in about 6 hours. 6 hours to 24 hours, when most TB phenotypic testing takes 2 weeks. Hey. Yes, the one at Denver, Jerry, Nick, and yeah, National Jewish. Yeah, absolutely. So using phages for Mycobacterial Research, my understanding though is you don't have a single cocktail of phages that you can give everybody. I think you have to look for phages specific to the strain. So there's going to be work involved in that. And I think all of this is experimental. I mean, that was incredibly successful. Like, you know, this is, so the case that we're talking about is a young man with CF who had really bad lung disease and had recurrent resistant CF. That he was going to National Jewish for treatment, and they used phage therapy not only to clear the CF infection, but to get him a lung transplant, which is a real level of stringency. You know, if you want to clear an infection, if you have somebody with Mycobacterium abscessus, you are not going to give them a lung transplant unless you're really sure. And it's a single case report, was published in Cell, but it's it's incredible. I think they had like 120 isolates over the course of his treatment, and they've done a lot of really interesting analysis with that. So we could use phages, but I think it's going to require a lot of work for each individual case. Oh, absolutely. Absolutely. That's a good way of thinking about it. Anybody on Zoom with questions? Oh, how do I? Oh, there we go. Sorry. And we can just end it. Let's just end it. Can we maybe move the mouse that side? Oh yeah, I can do it.
Oh,
yeah. You see a lot of Bayesian lineage. All right. So I think the first question from Tina is, did we see a lot of lineage too in India, and did it correlate with MDR-TB? So we didn't actually have phenotypic data for all the isolates. There is more recent data showing that, so Beijing sublineage is lineage 2, which affects East Asian. We saw a lot of that in Northeast India. The tricky thing about inferring whether it's more resistant is that there's a selection bias here. Most strains that get cultured in India are only in people who are failing treatment. And so when they've tried to link lineage with drug resistance, it's not really settled. Within Mumbai, which is sort of where you see a lot of drug-resistant TB, there was a recent paper showing that a lot of it is, again, lineage 2. But I don't know if that's just more transmission than anything else. All right, sorry, question, we can't hear the questions, so sorry about that. And so, Amanda, your question is mass spec diagnosis. It's actually mainly protein-based. It's not lipid-based. So the clinical test that we use is proteomics-based. And so Tina's giving us an update about the patient who I talked about. He doesn't have HIV, normal immunoglobulins, no diabetes. So he's a positive sweat test, so potentially has CF, and we're waiting for the CF gene testing. And then, is anyone doing lipidomic analysis for NTM? Yes, people are doing it. I think Colorado State is where they're doing a lot of this work. And people have actually shown that you can use MALDI-TOF to strain type as well. And differentiate between species, and in some cases, hopefully actually identify resistance. And then what is the status of vaccine development for NTM? I don't know, but the status of vaccine development for TB isn't going on, is in progress. So hopefully that is going to inform NTM. I think that it's going to be challenging with NTM if the genetic diversity of the strains are so much. I think we're at time. Thank you.